before I introduce Cristina, I'd just like to say Cristina is going to be the last of the talks of the candidates for the junior faculty position. And as you know, that besides meeting with the faculty members, all these positions have also been meeting with the students and postdocs. So what we would like to meet with the students and postdocs, and I'll arrange it in about probably about a week or two, and discuss to get your views on the candidates, okay? And it's going to be up to you to organize how you want to do this. If you guys want to get together, get someone, a leader, a representative who tells your opinions, or if you'd like to give us in a formal way in terms of rankings, or if you want to give us, just want to talk informally with us as a committee members. But anyway, I just wanted to warn you what's coming. We're going to be talking with you, and we're going to expect your input on the candidates, and it's going to come to a ranking. And this is true of the faculty too. In the next faculty meeting, this will be an item, and I'm going to get Mark to call one as soon as possible after this, so we can start moving forward. Okay, and so we're going to want rankings. All right. That being said, we'd like to welcome Cristina. Cristina doesn't need as much introduction as most of the candidates. Most of you know her quite well, particularly the ones here at the Institute. But you might not know a little bit about her past. She did a Ph.D. at UMass with Karen Zerpola.
That was undergrad in Brandeis. Jose Lemos was at UMass.
Okay. So she went on, stayed at UMass went to the Brudnick Institute to do a postdoc with Andrew Tapper, and then later, as you know, with Steve Treisman. She came to the Institute with Steve as a postdoc. Until 2014, she's made adjunct faculty in anatomy and neurobiology. She's consistently worked on the roles of voltage-gated and other kind of channels, mostly voltage-gated channels in drugs of abuse. Today, she's going to be talking about her work for the last several years in alcohol tolerance and PK. Well, thank you. I welcome you here.
Thank you. Thank you for being here. I know that it's been a rough week, lots of exams and all that. Especially thank you to the committee for extending an invitation to present the work. The title of my talk is Alcohol Tolerance, Making the Link from Molecular to Behavioral. So the brief outline includes an addiction, talking about addiction, the role of tolerance in addiction, different patterns of drinking, and the role of BK and its connection to both alcohol tolerance and consumption. We have several different projects going on right now in the lab, and I think I can divide them into molecular, behavioral, and transcriptional. On the molecular front, we have post-translational modifications, desensitization, internalization of the BK, and lipid RAF association, all of these with respect to alcohol exposure. More recently, we've incorporated behavior, specifically drinking in the dark, or DID assays, and this has been done by Roberta Ponte, which is an undergraduate student in the lab, and in collaboration with Dr. Gregory Quirk, we've been doing fear conditioning and extinction as well. A little bit newer and very much in progress is this transcriptional component, which is TCF, LEF transcriptional regulation, and also looking at differential expression profiles of microRNA in response to in vivo ethanol exposure. This has been done because now we have more and more facilities to look at microRNA, and with the COVID grant and Josh's involved, actually we've been able to do really some interesting stories about microRNA expression. So let's start by defining alcohol tolerance, which essentially is characterized by a reduced drug effect, where the effects of a drug decrease in spite of an altered concentration. This means that in order to get the initial effect, you need a higher concentration of alcohol. Now, alcohol studies are basically divided into three main factors. You look at direct tolerance, you look at the speed of recovery from tolerance, and the resistance to developing tolerance itself. Each one of these components fall into the three types of tolerances, which is the molecular tolerance, which we'll be talking about quite a bit, cellular and behavioral tolerance, which essentially is how difficult it is to actually show signs of inebriation, for example. Now, each one of these types fall into different exposure patterns. Acute exposure of alcohol, which is 15 to 30 minutes. Rapid in flies is defined as 4 hours. in mammals, anything from 1 to 24. And chronic exposure, which a lot of the research has been focused on chronic exposure, it's 24 hours or more of continuous or intermittent alcohol exposure.
When you say exposure there, does that mean how long it stays in the system too?
It's how long you've exposed the system to it. How long it actually stays in the system, I think it depends. For example, you have to get BCs to know how long it metabolizes for.
Yeah, just curious, how would you get like a 15-minute acute exposure in an animal? Is that possible?
No, I only heard of acute exposures in actual cultured neurons. Yes. So the association with addiction. Addiction is defined as chronic, relapsing, and this is an important point, it's a brain disease that is characterized by compulsive drug-seeking use despite harmful consequences. Addiction itself has 3 main stages, and this has been defined by Dr. Koop, who's now the director of the NIAAA over at NIH. And we have positive effects, which is the binge intoxication, euphoria, the pleasant feeling of being intoxicated. Then you have 2 components of withdrawal and preoccupation, which are negative effect. Now, the withdrawal will give you the preoccupation, the sweats, the nausea, and the preoccupation with increasing amount of consumption. Binge, we're particularly interested in because it passes to withdrawal via tolerance. So when you develop tolerance to alcohol, you need increasing amounts of alcohol to get the intoxicating effects, which leads to the next step in the cycle. Now, as substance dependence on alcohol progresses, what you find is that positive reinforcement, the euphoria, gets replaced as a motivator for drinking for the negative reinforcement, which is trying to avoid feeling the withdrawal symptoms and trying to avoid the anxiety of the preoccupation with drinking. So for our purposes, we're going to really focus on the positive reinforcement at very early stages of substance dependence. Now, if we're going to talk about binge drinking, let's take a minute to define it. According to the National Institute on Alcohol Abuse and Alcoholism, binge drinking is when a person drinks and it reaches a blood alcohol concentration of 0.08 grams %, which is mildly intoxicating or above. This typically happens when a woman consumes 4 wine versus a man who takes 5 drinks or more. Now, what's interesting about binge drinking is that 90% of alcohol that's consumed in younger people below 21 years is in the form of binge drinking. And more than half of all the alcohol consumed by adults is also in the form of binge drinks. Now, drinking too much, whether it's because you overdid it with a binge drinking or just because you are an alcoholic, causes losses in productivity, healthcare, crime, and other expenses, and that's the main concern of society and the NIH. And they've been able to estimate that about 77% of those costs are actually associated with binge drinking, which is around $191 billion. So my goal is to understand the central nervous system adaptations to alcohol exposure that underlie the development of alcoholism and the alcohol-related problems. And the big gap in the knowledge so far is that most of the research has been done on chronic or alcoholic translational models. And what I'm hoping to do is to understand the effects of more common drinking patterns, the non-chronic, and the central nervous system and how they can mediate tolerance and how that develops into subsequent decreases in consumption. It looks a little blurry, right? Okay. So these are the 3 stages of the addiction cycle. Each one of these stages is associated to specific circuits. For example, a preoccupation is the basolateral amygdala, insula, hippocampus context, meaning, if you walk into a bar, you anticipate drinking. And that's where the hippocampus comes in, prefrontal cortex, making decisions. Withdrawal is hypothalamus, brainstem, a lot of autonomic systems. That's when you get the sweats, the nerves, because you're in withdrawal. But we're going to be focusing on binge and tolerance. And that particular circuit really focuses on the striatum. So there's reciprocal inputs from the BTA to the nucleus accumbens, which is part of the ventral striatum, and that leads to dorsal striatum and directly to the thalamus. This is considered the direct pathway for reward. indirect pathway leads to globus pallidus and then towards the thalamus. And that's more associated with aversion, but it has components of reward. Now, going back to the main goal, which is linking molecular through behavior, all the way to behavior. Very few studies have actually been able to do this, and even fewer studies are relevant to alcohol tolerance. One particular study is really important. It's the Martin 2008 paper, which looked at the beta-4 subunit BK channel knockout. When you knock out the beta-4, what happens is that now the BK develops acute molecular tolerance. And that acute molecular tolerance, you can see the effects on cellular and behavioral levels, where the animals who are beta-4 knockouts drink more than their well-acclaimed counterparts. And using this as a model, I'm going to continue with wild-type, non-genetically modified animals to look at that link at different molecular tolerances. Now, the importance of the BK is not just circumscribed to mammals. We also have evidence of this in the invertebrate models, such as Drosophila, where the BK knockout shows a reduction in the recovery of alcohol sedation. And interestingly, the C. elegans, if you knock out BK, they develop resistance to ethanol-induced motor incoordination, i.e. they don't look drunk. So we're going to take advantage of the fact that BK is really interesting in the sense that it has a strong potentiation to BK at physiologically relevant concentration of alcohol. And it furthermore does develop different distinct forms of tolerance that we can really exploit to bring back to behavior. Now, you're probably all really familiar with BK, but I'm going to go over this fairly quickly. The BK channel has 4 subunits. Each one of these 4 subunits, the alpha, has 7 transmembrane domains, and it has a cytoplasmic tail. This channel conducts mainly potassium at very high conductances, and it's both voltage and calcium activated. Okay? It is modulated, like I mentioned, by beta subunits, which associate to the alpha. Now, what's really cool about the BK is that it's really essential in action potentials that are in the burst, neurotransmitter release, and also dendritic excitability. So it's a key target. So briefly, we've looked at in the past many different aspects of the BK, including how the lipid environment affects ethanol response, splicing of the channel, post-translational modification, and association with the beta subunit. We're not going to get to talk about all of those, but one of the techniques that we use in that, I was actually well-trained now on cell-attached or single-channel recordings of the BK channel. So essentially, you have a small little patch and you can see single openings in the channel. And there we can analyze the open probability in response to alcohol. So when I say that BK potentiates, I say that the open probability of the channel is larger. In other words, it's more likely to be open, larger opening times. Now, if we make a hole with complete access into the neuron, we now can listen to the summed effect of all the channels that are on the surface. And those channels now give you macro currents. And these are macro currents for different depolarizing steps of voltage. With those macro currents, I can look at current density, which is an indirect measure of functional BK channels on the surface. Now, why is this important, and how does this link? Well, here you have a medium spiny neuron. These are the types of neurons that you're going to see in the striatum, which is the circuit that we're particularly interested in. And the BK channel on the surface determines firing rate in a very particular way. So if I see the BK channel have an increased open probability, what happens is that that translates into a decrease in firing frequency. And the opposite is also true. A decrease in open probability leads to an increase in firing rate. So what we know from the literature in terms of alcohol effects, we know that both increased firing and decreased firing have been observed in the nucleus accumbens in response to alcohol. But of course, this is very region and neuron specific. So those constraints, that's why some of the data doesn't actually match. And another important consideration is the concentration of alcohol and the exposure times. So up to now, there's no cohesive understanding of how alcohol affects. So briefly, just to go back to the idea that behavior is well correlated with molecular effects of alcohol, I'm going to use it as an example, alcohol acute tolerance. Now, we know that that is characterized by a decrease in the open probability in the presence of ethanol. So you have the initial potentiation, and then it creates tolerance such that even in the presence of ethanol, the probability goes down to control. That causes initial decrease in firing, and then it returns to control levels. And furthermore, there's an increase in alcohol consumption when you have acute alcohol tolerance. This is the data at the molecular level, where you find that the open probability increases significantly in the presence of ethanol, and it stays up. Here, there's no acute tolerance for the alpha beta 4. But when you just have alpha, you have the initial potentiation, and that develops into acute tolerance, goes back to control in the presence of ethanol. In terms of firing, you see that the wild type fires normally, and then in the presence of alcohol, decreases firing, and the decrease in firing is persistent. While when there is no beta 4, you find a decrease in firing, then recovery. At the behavioral level, animals that have been knocked out for the beta 4 show almost doubling in alcohol voluntary consumption as opposed to the wild type. Now, I followed up these studies to further understand a little bit of how post-translational modifications could actually impact the initial potentiation response due to alcohol. And in the presence of the beta 4, and without the presence of the beta 4, you still have a potentiation in response to pKa, but the potentiation in the presence of beta 4 is much higher. So there is a modulation that's differential with respect to phosphorylation. And in fact, if you go ahead and delete the pKa site in the alpha subunit, and these are mutations that were expressed in hex cells, you find that the alpha mutated form does no longer respond to pKa activation, but the beta 4 does. And the presumption is that there's beta 4 phosphorylation sites that might be acting independently. How do these changes in post-translational effects affect the response to alcohol? What we find, these little squiggly lines here are actually representations of pK potentiation over time with response to ethanol. Is that pKa completely blocks the potentiation effect in alpha, but it has no effect on alpha beta 4. In fact, what we find is that the beta 4 association confers a certain regulation to CAMK2, such that when you inhibit CAMK2, you now get acute molecular tolerance. You go back to the striatal tissue, which actually expresses alpha beta 4, and you see exactly the same phenomenon. You see a sustained response to ethanol, and you see acute molecular tolerance developed in the absence of CAMK. The point I want to make with this particular slide is that post-translational modification is intrinsically involved in the expression of acute alcohol tolerance. And that sort of brings me to my next question, which is, what are the effects of chronic alcohol exposure on the channel? Well, in this case, chronic exposure is 24 hours of continuous ethanol exposure. This is the typical potentiation that we've been talking about. After 24 hours of chronic exposure, you find that it no longer responds to acute alcohol. Also, clusters that are usually seen with the PK are not formed, and the clusters that still remain have a lot less PK channels. And there's strong internalization of the channel as well. And this is all at very low concentrations of alcohol, 10 millimolar, 20 millimolar. Key point is you see both desensitization and internalization of PK channels present in the chronic ethanol. But here's the gap in knowledge. We want to be looking at binge drinking. Binge drinking falls into the category of rapid exposure. So nobody had really looked at that, and that was the question we wanted to sort of lead with. Sort of to spoiler alert, if you look at these exposure times, we found that up to 3 hours, there's nothing persistent that happens, but between 3 and 6 hours, there are persistent changes in the BK channel. And there's two in particular that I'm going to talk about: desensitization, and here we coin the term persistent molecular tolerance, or PMT, and we also see internalization, which is the second component of alcohol tolerance. Let's start with desensitization. So this is going back to Josh's question. We look at acute alcohol in a petri dish. So we looked at striatal, hippocampus, and HEC293 cells, safely expressing the human sequence of the BK channel. What we do is we just add alcohol 3 hours versus 6 hours, wash it, and either immediately record the response to acute ethanol or just put it in media, allow for different time of withdrawals, and then test for sensitivity of alcohol. So in vitro pre-exposure does trigger persistent molecular tolerance after 6 hours, but not after 3 hours. And here you see the data. If you leave it in 3 hours and you give it 4 hours withdrawal, these are openings of the BK channel. You see that when you apply ethanol, the openings increase, and in fact, the openings are longer. But if you leave it for 6 hours and you leave the same withdrawal time, 4 hours, there's no difference when you acutely expose alcohol. And even if you have a dish at 6 hours and you wait for 24 hours, there's no difference. So in summary, 1 in 3 hours recovers at around 4 hours of withdrawal, recovering the potentiation, but 6 hours never does. And we didn't really understand what that was until we started to look at the kinetics and the mRNA. And what we found was that the mRNA expression of the Strex isoform of BK was exponentially increased. So the Strex BK is completely insensitive to alcohol. So what's going on is that you're having a substitution of the BK isoform that is sensitive to alcohol for the BK channel that is not. So we tried to publish this in Neuroscience, and the reviewers came back and said, this is in vitro. What happens if you do in vivo exposure? So we had to go back and look at different ways in which we could actually do this by doing in vivo. So I designed intraperitoneal injections that would allow a BEC, blood ethanol concentration, for about 6 hours of the right concentration that would mimic what we do in vitro. And in consulting with Dr. Melendez, I didn't know how to explain this, and we came up with a term, C exposure. C stands for systemic episodic ethanol. So it's a single episode, it's systemic, and it's ethanol exposure. So when I talk about C, that's what I'm talking about. So essentially what it is a series of intraperitoneal injections of ethanol over 6 hours. The 3-hour exposure is done by doing 3 ethanol, 3 saline, and the control are all saline. And depending on how we space it or the concentration of these injections, we can get either 40 millimolar or closer to 20 millimolar. Okay? So C exposure has become our model for binge drinking, which is associated to moderate intoxication in humans, because it's about 0.09%. When we went back to Neuroscience, we have this data where we show 3 hours BEC, 6 hour BEC, and when we looked at the NPO of ethanol over control, it was remarkably in agreement with what we had seen in vitro, where you can see strong potentiation in naive, strong potentiation for the 3 hours, but a very reduced response in the 6 hour exposed animals. And they accepted it. That was very hard. So the next molecular tolerance that I wanted to talk about is the internalization, or the second component. We're looking at this component 2 ways, because it's a tricky component. We're looking at it with current density, and we're looking at it with imaging. The current density is essentially looking at the amplitude of the current, and we're dividing it by the capacitance. The capacitance is linearly associated to the area. So what that gives us is the number of functional BK channels on the surface. Okay? And we're doing also imaging. We want to actually see the BK channel at the membrane, and we use confocal microscopy and also TURF in collaboration with Dr. Judolski. Stephanie did a lot of work with this in hippocampus, and it was a very nice work. And for those of you who aren't familiar, TURF, what it does is it's able to excite an evanescent body at a resolution of 200 nanometers. Okay? So you have the dish right here, and the evanescent wave is right here. So what you're looking at is the membrane and 200 nanometers above. That's called the perimembrane region. If you change the incidence of light, you can actually excite the rest of the fluorophores throughout the neuron and get a bigger picture of what's going on inside the neuron. So when we did current density measurements in the striatum, we found two important points. One, there is in fact internalization of the channel that starts at 3 hours, but the effects at 6 hours seem to reverse after withdrawal with actually an overshoot at around 12 hours of withdrawal. The effects of 3 hours recovers, but there's no further effect. So now we know that striatum expresses both desensitization and BK channel internalization. In hippocampus, it's different. You get internalization, but the internalization is persistent. It doesn't change, it doesn't rebound, it stays decreased in response to alcohol. These are images of TRF. So GM1 is standing for the lipids in the membrane. And remember, this is the bottom up, so this is main membrane, perimembrane region. You have the BK staining of that same neuron. And this is the intensity profile, where red is really intense and blue and purple is less intense. So you see there's really strong staining at the membrane and perimembrane region. With ethanol, you see a marked decrease. When you quantify this on the surface of channels that are 6 hours with 24 hours withdrawal, 18 hours withdrawal, versus 6 hours of ethanol withdrawal, you see that there's still a very marked decrease in surface expression. Total expression is not affected, and this is done with Western blots, and this is a sample of the Western blot. So essentially, we're talking about a redistribution of channels, not a degradation. They don't disappear, they just get redistributed. So taking advantage of the fact that hippocampus is sort of a hallmark for learning and memory, right? We really wanted to make a correlate with alcohol tolerance in terms of it being a long-term adaptation, similar in many components to learning and memory. And the hippocampus is sort of the right way to go in terms of the different components. We know, for example, that long-term adaptations require protein synthesis. For example, short-term memory to long-term protein synthesis, LTP, early LTP to late LTP protein synthesis. So the question is, in the hippocampus, the redistribution of BK to persistent redistribution, does it require protein synthesis? We did a series of experiments, and this is again with current density, where these are typical currents in the hippocampus, different levels of voltage. With ethanol, you see those currents have diminished. But in fact, if you incubate with cyclohexamide, 10 micromolar, which is a general protein synthesis inhibitor, ethanol no longer causes internalization of the channel. And we were able to look at this with 10 nanomolar cyclohexamide, which has no effect on the current density of ethanol. But if you up it to 10 micromolar, we see that it blocks completely the effect. And in fact, cyclohexamide alone has no effect on current density. If you test hex cells, you see there's no internalization at 3-hour ethanol, the 6-hour ethanol, but cyclohexamide is only required at 10 nanomolar to cause a reversal or a blocking of the ethanol effect. Now, it's really important to look at the VHAF and the single channel conductance, that those have not statistically changed, in order to be able to interpret the current density as, in fact, changes in the number of functional channels. This is confocal imaging, so supporting the data with hex cells. Red is BK alpha, blue is the nucleus. This is the monochrome for the BK, and the pseudocolor intensity scan showing a large amount of BK near the membrane. Ethanol, this is the periphery of the membrane, looks at BK more internalized, and the presence of cyclohexamide, the profile is much more similar to naive, and the data shows this. Okay, so we have something that's being synthesized that is critical for internalization in the hippocampus, but we don't know what it is. So we had to look into how we would isolate de novo protein synthesized proteins during this process and how we could potentially identify them. And one way we did this was by feeding the cells metabolically altered methionine, so that every single protein that was being made during a specific period would incorporate that methionine. And in a copper reaction, we now had an alkyne that we could target. So by taking advantage of the fact that all of the novel synthesized proteins had this alkyne, we were able to isolate all of them. And we identified them using tandem mass spectroscopy. This was done in collaboration with RCMI Translational Proteomics. Dr. Loida Melendez is the director, and Dr. Juliana Perez is a collaborator. I don't intend for you to read this. I intend for you to understand that there were over 700 different proteins that were analyzed. Of those, a subset were actually increased in the presence of alcohol. And in particular, just want to show you here, clathrin interactor 1, which as experiments progressed, we really came to understand that it's likely that the endocytic pathway is through a clathrin-mediated process. What we did focus on was beta-catenin. The spectral count showed a significant increase with very little non-specific adhesion. And we have to confirm that with Western blot. We've done many, many of these. And with ethanol, we have about 162% increase in beta-catenin in response to alcohol. We repeated these Western blots with hippocampus and striatal tissue, and hippocampus actually shows the same increase in blocks with cyclohexamide and doubling the beta-catenin in the striatum as well. So at this point, I had to figure out what beta-catenin was because I wasn't really clear. And I found that it actually has a long history with the BK alpha channel. It's been shown by Lesage that it associates directly to the channel, and in fact, it does regulate surface expression. And not only directly through beta-catenin, but also through big-4 kinase, which is a regulator of the Wnt signaling pathway. And what I found further was really interesting because it became the basis for my R1. In breast cancer cells, what you see is that there's an increase in miR9, and that increase in miR9 regulates the expression of E-cadherin. E-cadherin is a cell-cell adhesion molecule. Now, when E-cadherin gets downregulated because miR9 is targeting the mRNA, what happens is that beta-catenin gets liberated and it's no longer phosphorylated in a way for degradation. It essentially activating the beta-catenin signaling pathway. So why was this interesting? Why is this the basis of the R1? Because they had shown already in a Neuron paper, Andres, that alcohol increases miR9. And I thought this was the perfect link from epigenetic changes to internalization. And those experiments evolved from there. Furthermore, in hippocampus, again, linking to learning and memory, it was found that long-term potentiation required Wnt release. So is Wnt beta-catenin sufficient and necessary? In fact, if you add the ligand Wnt, you see a very marked decrease in current density. And with ethanol, if you block the Wnt signaling, you find that you block the decrease in current density due to ethanol. So it's necessary and sufficient. And in recent experiments, we've repeated this in striatal tissue, and we have very similar effects. Just as an aside, IWP2 as a blocker, what it does is that it doesn't allow secretion of Wnt. So therefore, interaction between Wnt and the receptor is blocked, and it's blocking the pathway from the very beginning. So, reading the literature, I found two things that were important. Beta-catenin associates directly to the alpha, and it does so through the S10 region of the channel. In fact, there is a putative GSK site. And the GSK site, if you block it, you block beta-catenin association with the channel and also any ethanol effects on the channel. So this is shaping our hypothesis such that stable expression of the BK is associated to beta-catenin. GSK is phosphorylating both beta-catenin and BK. The phosphorylation of beta-catenin that isn't associated to BK or E-cadherin gets targeted for proteolysis. When the pathway is activated, Dishevel sequesters GSK. It no longer phosphorylates beta-catenin or BK. BK becomes unstable at the membrane, internalized. Beta-catenin gets accumulated and translocated into the nucleus, where it activates TCF-LAF transcription. So drinking. How does this all relate to drinking? This is a Ph.D. comments that I took the liberty to modify. Institute of Neurobiology study in progress recommended 100 bottles of wine to science. What we did was, using the C exposure, we followed that up by the drinking in the dark paradigm and wanted to see whether or not these mice had any changes in voluntary ethanol consumption. What we found initially is that using a single bottle of alcohol, presenting it 1 hour after lights out, on day 2, there was about a 20% increase in the animals that had been injected with ethanol, and that was persistent on day 3. So that was very encouraging. But we were concerned that day 1 showed no change and that there was an overall parallel trend towards downwards on day 3. So we did taste aversion test, and we found that indeed there was a taste aversion associated to ethanol injections that was not associated to saline injections. What we think is going on, and this is based on consulting with Dr. Melendez, he published a paper in 2012 which clearly showed that taste aversion is consistently present prior to developing excessive ethanol drinking in ethanol-dependent mice. Now, at the level of the circuit, if this is the nucleus accumbens, there's 2 types of neurons, D1 expressing, dopamine 1 expressing, and dopamine 2 expressing. Dopamine 2 activation is associated with an indirect pathway or aversion, inhibitory avoidance. Activation of the D1 is associated with reward. If you're activating all the neurons, you're going to have aversion and reward. And what they hypothesized in the 2012 paper is that reward eventually overcomes taste aversion and leads to excessive drinking. We switched over to the 2-bottle choice. It's more classical, and we were getting a much better sort of consistency in terms of consumption. Here you have the control mice, which were injected with saline, and experimental mice, which were injected with alcohol. The blue line is how much water they drank, and red is how much ethanol on any given day for each one. Here we're plotting alcohol consumption only. Okay? So the first thing that jumps out is that the animals that had ethanol injections started drinking much less than the animals that were saline injected. And that goes back to the idea that they have developed taste aversion. The animals that are controlled show no real statistically significant difference over time. However, the animals with the alcohol do show an escalation. And we realized after we did the experiment, we should have waited a little longer. And that's what we're doing now. We're extending the CID until 15 days. Okay, so this is the final part of the talk. And what we're looking at here is associated diseases to alcoholism. So what are the effects of alcohol exposure on PTSD? PTSD stands for post-traumatic stress disorder, right? You probably heard this from veterans. Essentially what it is that after a traumatic event, some people develop this, which means that a context, a sound, something that reminds them of that traumatic event puts them into an immediate panic. And that's considered within the realm of anxiety disorders. And what happens is that PTSD patients, more than half of them, actually have alcohol problems. And nobody really understands why. Research has been limited to very high concentration of alcohol or people who are already alcoholics, who have already developed the disease. Of course, we're bringing this back to our model, which is moderate anticipation, more common drinking patterns, and seeing how those affect the development of PTSD-like symptoms. One thing that became very interesting in looking at the literature was that redistribution of BK channels within the hippocampus is directly relevant to contextual fear conditioning and extinction. So we really wanted to further understand how this would fit our model. I really apologize for the resolution. It looks fine here. So for contextual fear conditioning, you place a mouse in a chamber that has a very specific context. Usually it's like white and black lines, and you give it a foot shock. The animal freezes, and this is what we're going to quantify. Later, 24 hours later, you put the animal in the same box, and the animal freezes because it remembers that context associated to the foot shock. Over time, what happens is the animal dissociates the foot shock from the context because it's no longer getting the foot shock. And that's called fear extinction. it learns to dissociate the fear from the context. And this is really important because this is the paradigm that is used to study PTSD. If a person has PTSD, they can't dissociate certain components or certain cues from fear response. So anything that talks about fear extinction deficits is harking back to PTSD-like symptoms. Now, in the case of contextual fear, we know it's hippocampus-dependent learning. And in the hippocampus, BK mediates fast after hyperpolarization and is mediating also learning. Contextual learning is both hippocampal dependent and learning tasks and requires changes in intrinsic excitability. Intrinsic excitability means that the neuron is more excitable. In other words, if you have internalization of the BP channel, you're going to have less after hyperpolarization, and you're going to have a higher frequency of firing like we talked about briefly before. I'm going to show you that more clearly now. So these are after hyperpolarization thoughts, the peak after hyperpolarization and slow. What's an after hyperpolarization? This is a burst. This is what happens immediately after the burst. You see that it repolarizes, overshoots, and then comes back. This is the after hyperpolarization. When it's paired, when you have the switch off in the context, there's less of that, allowing for it to repolarize more quickly and allowing it to fire more quickly, okay? And that's BK mediated in the hippocampus. If you have less BK, you have less of an overshoot, and you're most likely to be ready to fire again. And here you see the phenomena in terms of firing frequency. The tone in this case is what they're using for a control. There's less firing frequency, and when it's paired, there's a higher firing frequency. The key point that I want to bring here is that, right, we're talking about BK, we're talking about expression of BK, and we're talking about firing frequency, but what's key is that in order for extinction to occur, this McKay paper, 2009, clearly points out the fact that these changes need to be reversed in order for fear extinction learning to occur. Okay? Now, we did some preliminary experiments with Dr. Gregory Kirk. We're using our C paradigm, and these injections are occurring in day 2. The red line here in the conditioning day 1 are the animals that are going to get ethanol injections, and the blue line are the animals that are going to get saline injections, okay? So after conditioning, and this was both tone and context conditioning, they received the injections, and on day 3, we did an assay for extinction. And what we found was clear deficits in extinction learning in the animals that had received ethanol injections, right? And this was persistent during retrieval. So this has components and deficits of both hippocampal learning and basolateral amygdala. If you put this in the bigger picture, when you have a traumatic event, one of the first things you want to do is go out and have a drink. And you don't necessarily have to go all out, but some people have lots of drinks. And what this potentially identifies is a section of the population that could be at risk for developing PTSD and future alcohol abuse and dependence because it becomes a cycle. As the PTSD or anxiety increases, the more alcohol you consume, and hence the co-occurrence that we see in clinical literature. So our hypothesis here is essentially, when conditioning occurs, you have the changes in intrinsic excitability that occur in the hippocampus. With ethanol application, if you remember, there's a persistent change in BK channel surface expression. So essentially what we hypothesize is that ethanol is sort of locking in BK so that it can no longer reverse the intrinsic excitability changes during extinction, i.e. causing extinction deficits. Okay, so future experiments. This summer, my student Roberto Ponte is going to go to Mount Sinai, and he's going to do some experiments with Dr. Eric Nestor. And what we're hoping to do is really target beta-catenin, the regulatory pathway for this internalization, using pre-loxed mice. We're going to use stereotoxic injections, the nucleus accumbens specifically, and then look at that 2-bottle protocol for GIG specifically. I do want to congratulate Roberto on getting into the prestigious Icahn School of Medicine summer undergraduate program, and hopefully that'll be a budding collaboration that would give good fruit in the future. So briefly, we found that 6 hours exposure causes tolerance. Desensitization and surface expression changes, those are regulated with signaling. In terms of translational, we know that it actually affects the way these mice consume voluntary alcohol. It both facilitates it and it causes rapid escalation, which we're now working on a little bit more. And finally, as far as co-occurring alcohol diseases, we found that PTSD-like symptoms are actually triggered by the C exposure that we've been characterizing. So a take-home message, not to sort of spoil the party, but binge-like alcohol exposure can potentially elicit persistent components of alcohol molecular tolerance. And this could be facilitating alcohol consumption and alcohol-related diseases. And this is something that we're working on to be able to sort of tease out a little bit more carefully. I want to quick thank a very talented group of people. So these are my 3 undergrad students, Alexandra Burgos, Catherine Cordero, Roberto Aponte, which I already mentioned. My lab manager, Jessica Soto. 2 collaborators, Dr. Garcia and Carolina, and Dr. Marrero. So Dr. Marrero did analysis programs for both single channel and whole channel analysis, and Marla Rivera in Dr. Diaz's lab was actually really important in sort of working out the quirks of those programs. Dr. Garcia does discontinuous sucrose gradient, which I didn't have a chance to show you the data for that. Alexandra Burgos is wonderful at Western blot. She's actually pioneering assay for Wnt secretion and miR9. Jessica Soto is looking at TCF, LEF, OSET, miR9 isolation. In imaging cultures, I want to take a minute to acknowledge Stephanie Palacio and Garrett Seal, which work with the hippocampal prep and the imaging. Jessica Soto and Alexandra on the confocal, capturing viability assays. Behavior, like I mentioned, Roberto and Roberto Menendez. I had very helpful discussions with Stephen Trisler, Guillermo Julowski, and Carmen Rodera, which is a postdoc in Guillermo's lab, was extraordinary in troubleshooting Western blots. Judson Chandler, and very special thanks to the Colbert group. The directors, Joshua Rosenthal, and every Friday the group sets together, and we really have to work out everything, and we have to discuss it, and we have to understand it, and we have to analyze it, and I can't tell you how immensely valuable that's been. Discussions with Jorge Miranda, Manuel Diaz, Bruno Mani. I don't only have collaborators. I have a collaborator in UMass, Jose Lemos, Gregory Quirk at the Medical Sciences Campus, Eric Dessler, Mount Sinai, Leva Trump at Yale University. And finally, the next generation. So we're training the next generation of undergrads that are going to be substituting the current generation that are going to be moving on. We have Jose Ojoa, which is here, Charissa, Jimenez, y Ernesto Hernandez. With Ernesto, we actually are learning something really interesting, biosensors for neuronal signaling with Samuel Martinez and Jose Peñalver. So thank you very much.
So, a question about how do you, when you justify the appropriateness of the fear conditioning to PSD, PSD seems something that's very, very persistent and doesn't go away, whereas the fear conditioning model goes away in the matter of hours. I'm not in this, but I'm just curious how you guys, when you make a connection like that, how do you justify it?
So the question here, if I understand correctly, is essentially, if this is PTSD, it should essentially be more consistent here. It's sort of going down, right? Essentially, this is the model that they're using, but in fact, I agree. I think that in some cases where the deficit is actually, in fact, very severe, you have no extinction learning. In this case, you have a deficit. So in the typical model for PTSD, you might have animals that just simply don't.
These models, if you adjust, I don't know if people do this, they adjust the intensity of the shock and stuff. Is that
yes
at all?
I haven't looked at the different permutations, but every one of these subsections have been done very differently. Sometimes they wait 48 hours, sometimes they do it right away. In fact, they have tried, this is not novel, trying to see what ethanol does. They've tried to do this before conditioning, a single injection, and after conditioning, a single injection, and nothing happened. So the levels of variability that they've tested are considerable. We just happen to hit the right dose and time.
Yeah, Cristina, a lot of people think that GABA A is very important for alcohol. So I think to see if it's involved in anything. So.
I have not, but many people have. And the reason why GABA is really important is because a lot of the members of the circuit of the board are actually inhibitory neurons, which are GABA neurons. The research has shown, in fact, if you target those neurons by using GABA inhibitors or activators, you can modulate the amount of cravings and the amount of regulatory. Here, I actually have a slide on that because I thought it was really important. Here, baclofen is a GABA inhibitor, and it actually decreases alcohol cravings and manages withdrawal symptoms. So it is a really viable target for alcohol-related problems. I haven't done anything on it.
Do you know if the cells that you work with have got GABA A receptors on them?
They have, I'm not sure. They're GABAergic, but I'm not sure if they have GABA A receptors.
Because the medium spike neurons in the striatum are, most of the population is GABAergic, so
absolutely,
the right pathway, they should be like gatekeepers regarding some
absolutely, but I think your question is whether or not they're expressing GABA A receptors, right?
Well, GABA A is a big alcohol target, so I was just wondering how much whether some of these effects might be going through GABA A or if they're all just there's a direct. It's just direct through the PK channel that GABA is involved.
I think the PK channel simply modulates how much GABA is being released. So in terms of binding frequency.
So do you think the alcohol binds directly to GABA through PK?
There is actually a paper by Alejandro Dopico which shows that there's a pocket in the C-terminal domain that does directly associate to alcohol and modulates the channel that way. So there is evidence for that. But in general, the literature doesn't have like a receptor for alcohol. Alcohol has incredibly amount different effects.
So, I mean, you can imagine alcohols being fairly non-specific. How specific is it for PK versus, you can change excitability by changing 100 different channels in the system?
And in fact, it's absolutely true. In fact, while BK is being downregulated in the neurohypophyseal terminals, we have calcium, both the calcium channels that are being upregulated. So there is a lot more going on in this story than just BK.
So why do people think that BK is one of the major ones?
Well, the reason that I think essentially goes back to the genetic data. When you go back and you knock out BK, you have really clear profiles for behavior that is associated to alcohol. That isn't necessarily true for knocking out other potassium channels.
How do you explain alcohol avoidance in the choice test? We've had a minimal exposure. And where's the association of the exposure to the effects of the avoidance?
Okay, so let's go back to that really quick. If I understand the question, you're essentially asking me why it's developing avoidance?
If you have a short exposure to ethanol, then you have given two choices, water or ethanol. You're saying that for those who have been treated with ethanol, avoid substrate.
Right.
So there has to be some kind of association, and that seems like a rather rapid time in which we develop an association.
I think the association isn't necessarily taste. I think it's at the level of the actual circuit. So when the circuit receives a certain amount of alcohol, that's what it's associating to an inhibitory avoidance.
But then we're talking about periods of seconds to trigger this avoidance.
Trigger the avoidance is actually about 30 minutes to an hour.
So they drink,
They taste it,
and
and then they don't want any more to do.
But during that hour, they make it back.
They actually don't.
Which
So,
means it must be actually much, much more bad.
so within 30 minutes, I think it would actually reach the circuit. I'm understanding you're saying there's no, they can't make association behavior-wise because it doesn't take enough time. That's an interesting point. I'm not sure if 30 minutes is enough.
That's what you saw.
That's what we saw. And we think it has to do with the circuit, not necessarily with correlating behavior or the actual drinking with it. It's the system that sees behavior that's already tuned to an inhibitory avoidance response that is seen as a taste aversion. It no longer wants to have more alcohol. Taste is probably the wrong word, but that's what the assay looks at. What it's actually doing is telling you whether or not it wants to drink.
What is interesting for me is that the shock is a few milliseconds, whereas the alcohol consumption is much longer. So here you're getting one basically extremely rapid version, fear, version extinction, but then we've got very different responses.
Yes. So now you're talking to me about this. This is 24 hours after. Okay. And it's the same paradigm. And you're seeing the effect throughout the third day, 24 hours after. So I'm not sure what the question is. Sorry.
So they drinking alcohol or is it injected?
The C exposure is injected. So the idea here is
So
to,
they wouldn't taste it.
no, not here, but I think,
The other one they tasted. What's it taste of? Is it? It's not flavored, is it?
no, it's not. It's just
It's
alcohol.
just alcohol.
So here, for example, they taste it. And you can see there's a very big difference between the initial consumption and the control versus the ones that have been injected with ethanol. And we're trying to understand why is this occurring.
Right.
And
You
what,
wouldn't know what it tasted like if it'd been injected with it.
no. And that's, I think, what Dr. Cooper
Yeah.
is making in terms of the point. But what we do know is that this taste aversion to alcohol is actually consistently present in animals that later on go on to drink. So how this actually works, nobody knows. One hypothesis is what I had already mentioned, which is activating both inhibitory avoidance and reward, and that eventually the reward pathway overcomes the inhibition.
I think you showed a video right where you made mechanical neurons to after the 6 hour exposure. You didn't see the downregulation of the BP, is that correct?
Yes, that's when we used 10 nanomole. Is that what you're?
Yes. So do you think that, why do you think that's the case? And second, do you think that perhaps the DK channel may have more of the isoform of distress, which are not sensitive to alcohol exposure?
That's an excellent question. I actually had to address that for the R1. And when I looked in the literature, there is actually present a different protein synthesis inhibitors passing through the membrane differentially. So it might be a factor of permeability. So the membrane for the hippocampus is different for the hex cells, and one possibility is that that's an issue. And in fact, we know that some of these protein synthesis inhibitors can't get through to the nucleus or to other subcellular regions because they're not as permeable.
And then, do you see, what do you expect to see after the 6 hour of binge drinking and upregulation of the channel during recovery?
That's a great question. And I'm still, you're talking about this one, right?
Yes.
Okay.
Yeah.
So she's talking about the fact that there's a rebound here. And if we look at this in terms of the circuit and in terms of firing frequency, one hypothesis is that the initial downregulation of the BK entrains the reward pathway. And the overshoot of the BK now makes it harder to activate or increase an intrinsic celibate. And that's sort of the key component for tolerance. Now it requires more alcohol to get the initial effect. But that's just a hypothesis. We don't really know. This part of it, we haven't been able to relate back.
Are you trying to relate your Strex alternative splicing with your mRNA work in any way?
A little bit, yes. In fact, what we're doing with the mRNA is that we have two constructs. We have the 0 with a 3' UTR and the Strex with a 3' UTR. And those have specific sequences that adhere to microRNA. And we found many microRNAs that are associated to that. Specifically, miR9, let's use the example. But in fact, it has more affinity for the mRNA for the 0. It has a 2.1 than for the Strex one. So even if it had the same sequence of adhesion, the initial sequence of the actual protein seems to affect at the mRNA level whether or not it gets targeted microRNA. So Strex being less targeted than 0.
When is Strex, when does it come into play? You mentioned that, but can you repeat that again?
Sure. So Strex comes into play actually potentially here. But we see it in persistent molecular tolerance more clearly. When after 6 hours, you don't see a response to acute ethanol, even though the channels are there and everything is working out nicely, what we think is happening is that this channel is not the same as this one. This is your 0 channel, which is sensitive to alcohol, and this is your Strex PK channel, which is insensitive to alcohol. And that's how we're getting this persistent non-response to acute ethanol. Does that answer your question? Thank you. Thank you so much for your time.
Thank you very much.